Today I'm going to tell you about how to evaluate your health based on your stool sample. It is not a novel topic, but I would like to show you how to do it from a slightly novel perspective. So before we think about how to evaluate our health from stool, I would like to quickly tell you and convince you why it's worth evaluating your health from your stool. I'm sure that most of you have heard of the gut-brain axis, which is this beautiful phrase emphasizing the importance that our gut has on our body. It remains a chicken and an egg question whether it is the microbiome that is changing in disease or whether it is the disease that is changing the microbiome. But we do know that the microbiome does change in disease, and we can use this information to diagnose. Microbiome-based diagnostics are actually becoming a very interesting alternative to currently used methods such as blood collections or biopsies, because, as you know, they can be collected, microbiome can be collected regularly, non-invasively, and also thanks to developments in next generation sequencing, it's also becoming affordable. There are a few methods that currently allow you to estimate your health based on your stool sample. The first class is based on alpha-beta diversity, with the assumption that if your microbiome is richer and at a similar level of richness to the richness in microbiomes of other healthy people, then you're also considered healthy. The other group of methods is based on taxonomy. Those are slightly more complicated, but they still rely on a relatively simple premise, where they measure the ratios of good versus bad bacteria. And this is exactly what the Gut Microbiome Health Index, or the GMHI, and its successor HiPCA do. So one could say those are the gold standard methods for now. But why do we think that we actually need a new index? Let's have a look at the recently redefined microbiome definition, where we can see that the microbiota, so the taxonomic information, all the microorganisms, bacteria, archaea, viruses, fungi, they actually form a relatively small part of the microbiome. And it is the so-called theater of activity. So all of the structural elements, the metabolites that those bacteria produce, the environmental niche that they're present in, and even the interactions that they have with one another and with their host, that actually form the majority of this microbiome definition. This means that if we want to understand what's going on in the microbiome, we really need to take those other features into account. I'm also showing you here another reason for why I think we should focus on functions rather than on species alone. What you can see here is distributions of species in blue on the left and functions in pink on the right across healthy samples in a human microbiome project too. So we can see that when it comes to species, actually the majority is found in just a small percentage of healthy people. When we look at the functions, on the contrary, we can see that there are quite a few functions, actually a majority of functions that are found in almost all samples, right? So, of course, this was something that we only saw in the Human Microbiome Project too, but we also wanted to see whether it holds true for other cohorts. And this is the histogram that you can see below. So despite adding other cohorts and from different geographical locations and studies, we could still see a similar trend where there was an enrichment of functions in a feature called core functions, which were highly prevalent regardless of sample origin. So by continuing our exploration, we defined a set of parameters, which were features describing microbiome health. We then pass them to a random forest predictor, and we output a health score, which is basically our index, right? The probability of a sample being healthy, score of 1, versus disease score of 0. So we focus specifically on the IBD, or inflammatory bowel disease classification. And what you can see in the top, the big plot is showing the results of our index, and then the small plots are showing the results of the competitive indices. There are 3 cohorts that we are testing, and people who are healthy are shown in green, whereas the other colors are showing the 2 subtypes of IBD and some obese individuals. And the general takeaway is from here that our index was able to produce relatively consistent values for the healthy people and produce this nice distinction between people who were healthy and people who had different diseases. Whereas if you look at the results from the other indices, it was true in some cases, but it always wasn't. It wasn't always the case. Sometimes there would be no separation, or even there were some cases in which the indices were producing the opposite values. So actually giving higher scores for people who are diseased rather than those who are healthy. We also added some accuracy measures. We also passed the values of the indices as machine learning features to random forest model to see how important they were in the prediction. And overall, we could see that our index, here marked in red, had the highest accuracy and also seemed to be the most informative when performing this prediction. So as I mentioned, we only focus on the IBD individuals, but actually during the review stage, we were asked by the reviewers to see whether it worked on other diseases. And this was really interesting because, as I mentioned, we didn't think about other diseases at all, whereas the other indices actually were familiar with the remaining diseases that we used in our test cohort. And even despite this, our index seems to have, once again, the highest accuracy and the AUC in different data sets and different diseases. And the matrix I included here was, I'm not going to go into details about this, but I think it's just really, really interesting because what the matrix is showing the importances of different parameters that form our index. Those are related to different kinds of one could say different profiles of dysbiosis, and then there are different cohorts with different diseases in the rows. And different colors show you how important each of the parameter was. And what you can see straight away is that different diseases have different parameters as the most important. We suspect it is going to be dependent on which features of the microbiome are going to be distorted in what disease, and also whether the disease originates in the gut or whether it originates somewhere outside of the gut. But this is something that we are currently investigating, and we haven't managed to dig deep into this. Finally, I would also show you how well our index is able to produce the results regardless of sequencing depth. So the values of the index, of our index, are shown on the top right, and then the GMHI is on the left. What we did here is we took healthy samples and we verified them to different sequencing depth, right, to simulate different sequencing depths. And we can see that regardless of the sequencing depth, our index was producing consistent values and always giving the same classification. So the orange line is showing you the threshold for health. Whereas when we look at the GMHI, not only the values are not consistent, but also what is really worrying is that the GMHI actually changes the prediction depending on the sequencing depth, right? So this is something that I think is really important, especially if we want to unify the values across different cohorts with different sequencing depths. So in summary, we developed a method that is based on functions describing microbiome health, focusing on functions and interactions. We've developed this method on healthy versus IBD individuals. However, it's turned out to work really well for other diseases as well. Intriguingly, actually even better than taxonomy-based indices in some cases. This work is published in Giga Science, so please feel free to have a look at it. And I would also like to mention here that actually we are now working on the second version of the health index on a large data set of Polish cohort. So we would like to make this index very specific to the Polish cohort to aid diagnostics and maybe even treatment. We are thinking about probiotics specifically for the Polish population based on those functional assumptions. And this is done in collaboration with a Polish startup called One Biome, which does exactly this. So evaluates your health based on your stool sample and pairs you with a dietitian and a medical person to guide you in your lifestyle and improve your general well-being. And I would like to thank everyone who has been involved in this project, of course, my PI, Dr. Paweł Łabaj, but also everyone else. Thank you.